Question number 7. So this deals with the differences between genomic clones and cDNA clones. So remember that cDNA clones are basically a DNA copy of a messenger RNA. So really the differences here are what sorts of information are in a genomic clone, a genomic piece of DNA, versus a cDNA clone, which is made from messenger RNA. So if we think about what's in a DNA sequence, and we think about a typical gene, there's a lot of things. So there are exons and introns. So exons. There are also introns. Somewhere in here will be a translational start site, an ATG. Somewhere down here will be a stop codon, something like a TAG. And then there's also going to be a promoter, which is responsible for making sure that this gene gets expressed in the right places at the right times. And then furthermore, there can be other things like enhancer elements, which can be located upstream or downstream of the genes and are important in making sure that these things get expressed at the right times and in the right places. So whenever this is transcribed into an RNA, initially it's made into an RNA molecule that contains both the introns and the exons. But then this is going to get processed to remove those introns and add a poly-A tail to give you a mature messenger RNA. Now you'll notice, just generally speaking, there's a lot of things missing here that weren't up here. So one of the big things we think about are introns. So introns have been spliced out, so this is now gone. But also these regulatory sequences like enhancer elements and promoter sequences are not going to be present in the final messenger RNA. So with all that in mind, we can kind of go to the question here. So which mutation in this gene could in theory be detected with a genomic clone, a genomic piece of DNA, but not in a messenger RNA? So a point mutation that changes a single amino acid, so that's something that would occur in an exon, in a coding region, which would be in the messenger RNA. So we would be able to see that with a genomic clone or a cDNA clone. A mutation that prevents removal of an intron, well, if you prevent removal of an intron, then it would still be present in the messenger RNA, so that would be fairly obvious. A promoter mutation that prevents transcription. So this is the one. So those promoter sequences are not in the messenger RNAs. So that's something that you would not detect in a cDNA clone that you would with a genomic clone. So 10 base pair deletion in an exon. Again, the exons are present in both the genomic clone and the cDNA, so that could be detected either way. And a frame shift mutation also. That's something that's going to be happening in an exon, which would be present in both. So what you're left with is C.